everyone. So, hello and a very good afternoon to all of you all. So, thank you for joining us today in this particular webinar. So, this webinar is actually co-organized together with Invivogen. So, Invivogen is actually a manufacturer of life science research products. So, my name is Irene, and I'm the Senior Application Specialist for Apical Scientific Sri Berhad. So before I start this webinar, so allow me to give you some housekeeping announcement. So all attendees will be muted throughout this particular session. So if you have any questions, there is a tab present on the right hand side. So basically you have a view mode, so you can actually click on that particular question. So if you're participating using your laptop or your tab, so there will be a question tab, so you can just click in there and you can type in all the questions. Now if you are using handphones, so there will be an icon of question mark, right? So you can just click on that icon if you have questions, so you can just put in your questions. Now as for all the questions that you are going to put up in a while, so these questions will be answered through emails. So we will not be answering the questions here. So they will be all sent to you through a private email to your emails. So we have your email address as you have registered to participate. So we will send you personal email for all the questions that you have. Right. So without further ado, let's begin with the webinar. So today I am going to be sharing on the culprits, basically the unwanted cell culture contaminants. So before I proceed to today's webinar, So before I proceed to today's webinar, allow me to introduce Invivogen. So Invivogen was basically funded in 1997 by university researchers. So since then, what happened is they have gradually expanded. So they have gradually expanded with the opening of the San Diego, which is this. San Diego in 1997. So here you have here, and in Hong Kong. So they started off in they started off in 1977, and then they have moved. They have expanded in 1997, and again they have another one more sometime in 2014. So this particular company is actually known for its effective Microplasma detection as well as removal agents. Okay, so Invivogen is only is the only manufacturer for zeocin in the world, and this product has been used. Okay, they have used and they have packed by different brands across the world. So they are actually known for their experience and confidence in their antibiotic quality. So now let's move on to the next scope. Right. So basically, in this particular scope, what we are seeing is actually the introduction. What some of the scope that we will be covering up today. So we are going to see on introduction. We are going to see some types. Oh, give me a moment. Okay, so this is the introduction. So this is the scope. So basically we have the scope for today. What we will be looking into some of the introduction, type of contamination, how are we going to overcome contamination, and the conclusion. Okay, let me start with introduction. All right, so microbial contamination in cell culture is basically serious, and it is very much, it causes much threat in, especially for research. So it can range from a minor annoyances, so in which we can see this contamination. Sometimes they tend to reappear again and again, though much effort has been taken to curb this particular contamination. So we tend to have this over and over again happening. So this particular contamination can also cause a major problem, in which it can lead to disastrous result, loss of time, and a lot of resources that is being wasted. Right? So therefore, what are we going to look into this particular webinar is, we are going to look into how to detect, how to prevent, and how are we going to eliminate. So we are going to look into these three main sections together with some of Invivogen products. Okay, so microbial contamination usually or must be detected as early as possible. So this usually depends on the nature of the microbe itself. So some of the detection methods are like, you can use a biological assay, PCR. It could be a fluorescent or chemical staining. You have optical. So these are some of the detection method. And most of these detection methods, they tend to only be pretty much suitable for bacteria and fungus. All right, but this is not the case for Mycoplasma, because Mycoplasma usually can go unnoticed. So basically, because we cannot detect Mycoplasma visually, so that is why they can go unnoticed for a very long time. So the image at the bottom shows a Mycoplasma free cells, and that is been contaminated with the Mycoplasma cells. So where do we start? All right, so it is always by identifying, or the source of contamination. So just like the wise man used to say, prevention is better than cure. So knowing the source, so when you know the source, you can actually minimize the risk contamination. So how are we going to do that? So basically, the first most important thing is, you should know, or you should make sure that you are working in a proper sterile environment, and you are using a proper aseptic technique. So this is the aseptic technique. You must make sure it is all done properly. And then second is to quarantine any new incoming cells that you purchase. It could be from your very specific vendors, or you would have asked from your from the neighboring labs. So all this is good to get into quarantine first, so you can use a separate incubator. So you want to verify that whatever cells that you obtain, they are actually free from contamination. And finally, always observe and monitor your cell culture on a regular basis. So you can always do this. How are you going to monitor? You can always monitor using optical microscopy or any detection kit. So if you think that your lab is very much prominent or easily what we call this, gets into contamination, so detection kits is a must. All right, so typically, so once microbes are detected in the cell culture, so you need to, it is recommended, okay, to discard the cells. So the moment it can be any bacteria, fungal, or mycoplasma. So once they are detected, the best is to discard. But then there might be some cell cultures that are very precious, right, that it can't be lost or thrown, or right, easily be discarded base. So example, if you tend to have a stable clone selection, maybe any cell line, okay, that is derived from any explanted tissue, or even any primary cells. So these cells are very much precious because once you have lost, there is no, you don't have the resources to get back the same cell lines. So at such, how are we going to recover such cell lines is to use antibiotics. Okay, now antibiotics can only maybe bacteria and fungi. So how about for the rest? So now this webinar is going to address not only for bacteria and fungi, but we are also going to look into how are we going to detect, prevent, and eliminate those that is very difficult to be removed used by using antibiotic, for example, like mycoplasma, endotoxin. So we are going to see how we are going to detect and eliminate them. So now let's look into the most common source of microbial contamination in lab. So the first thing is infrastructure. I mean, where are you doing your cell culture work? So your fume hoods. So you have to make sure your fume hoods are well ventilated, okay? And then your areas that you're working is basically cleaned, right? So this is the infrastructure. Operator is those who are the laboratory staffs. Okay, so you want to make sure that when every time you're doing a cell culture work, that you are wearing the proper PPE as well. All right, and you want to practice a good aseptic technique. Now, pipettes, tips, syringe, as well as vacuum pumps, they can also be the cause for contamination. So this is also you need to look into. Equipment, safety garments, culture media reagents, and cells are also could be the common source in the microbial contamination in your lab. So these are some of the common sources. So now let's look into the first contamination, which is the mycoplasma. So we are still going to look into how are we going to detect, all right, how are we going to prevent, and how are we going to eliminate this. Now, mycoplasma is it's known to be the most frequent contamination in cell culture. Okay, so mycoplasma is the smallest and simple self-replicating organism. So this mycoplasma is basically, or they're relatively small with no rigid cell walls. So that is why it becomes a bit more difficult to detect through visual inspection. And not only that, they are usually able to pass through standard filters, and they are also resistant to a large number of antibiotics. So mycoplasma are also high infectious towards all type of eukaryotic cells, including the primary cells. So it has the ability, right? It has the ability to attach to a single cell. All right, it can attach just to a single cell. It can fuse to the cell membrane, multiply, and they can outnumber the cell culture by 100-fold. So that is why drastically it tend to alter the cell culture, itself, and a lot of time it tend to skew the research results. So this is actually an infographic to show mycoplasma in number. So being identified in 1956, okay, there are about 180, or could be more, there are more, different type of mycoplasma species that has been identified till today, okay. And out of these, there are 6 species. So if you can see 6 species here that are identified, which are known to contribute about 95% of cell culture contamination. All right. Now, and surprisingly, 80% of this mycoplasma contamination are usually from the lab operators. So 80% of lab operators are carrying the mycoplasma. So we can see that this is the main contributor for Mycoplasma contamination. So that is why it is good that we tend to prevent them before looking into cure. All right. So what are the major impacts? So the moment your cell culture is infected with Mycoplasma, what does it do? So it can actually alter the cell function. All right, so it can alter the cell function. So basically, these are some of the cell function that the Mycoplasma gets to alter, and eventually it all leads to cell death. All right. So Mycoplasma competes with the host for nutrients and biochemical precursors. So in the image, you could it tend to alter the metabolism of the cell growth and cell genome. Not only that, it tend to damage the cell membrane, disrupt the cell integrity, interfere with membrane receptors, example like ion channels, okay, that it can further cause damage to the cell, whereby they can interfere with the signaling cascade, cytokine production, right? And also components like cytopathic effects, which literally brings to the cell death. All right. So these are the common source, all right, of Mycoplasma contamination. Okay, this is the common source. So usually incoming cells, all right, contamination coming from the other research laboratories, lab operators. Okay, if your water bath, incubators, or glassware are not well clean, basically they tend to be dirty or not autoclaved well, all right. And then you have biological reagents. Sometimes it's just sometimes that even these biological reagents can also carry Mycoplasma. So always ask for certification, okay, to prove that whatever biological reagents that you're buying from the respective company, they are free from Mycoplasma. Okay, so now how are we going to detect? All right, so the first thing to you can use to detect is using plasmo test. All right, so this is the first there are few detection kits. Now this is the most how to say, I would say that very much commonly used. All right, so to detect the presence of Mycoplasma contamination in cell culture, so Invivogen has actually introduced a colorimetric cellular assay. Now, which is actually sensitive. If you can see, it can detect quite a large number of these particular Mycoplasma in the your cell culture supernatant. Okay, not only that, it is reliable, rapid, and it is also a simple way, because the hands-on time, right, it is basically less than 1 hour, and you can just get it after an overnight incubation. All right, so basically the procedure is also same. So if you see my cursor, I've moved here. So all you have to do is just collect and heat the cell culture supernatant. All right, you just add in the HEC blue 2 sensor cells. All right, you just have to add in the, it's a detection medium, so you just put in your cells. You can put in this particular detection medium, you incubate overnight, and if this particular culture cells is Mycoplasma, if there is a presence of Mycoplasma, then it will be purplish blue. All right, if it is negative, then you will get a pink. So basically you can also measure the OD between this. So if you have your plate reader, you can also use your plate reader to identify them. All right, so why you are encouraged to use Plasmo test kit? Because it detects all Mycoplasma species responsible for cell culture contamination. All right, and a positive result indicates a contamination. So if you have this particular solution turning into purplish blue, then there is presence of Mycoplasma. All right, and then you can actually perform them in 96 well plate. Okay, and then when you purchase this, you can purchase this kit and you can freeze them. You can actually freeze these particular blue HEC blue 2 cells, and you can reorder the refill and other components separately. Okay, so if you see here, we have the test, we have the controls, and you have the refills. So each of these have, you can basically, the controls can go for about 200 tests, refill for about 500 samples. So when you buy this test, you have one kit that can be used for 250 samples, right? Now, Plasmocine, this is frequently cited as the mycoplasma removal agent. Yes, okay, so it's actually frequently cited by many journal papers. All right, it is effective against all common mycoplasma strain, both extracellular and intracellular. Okay, so this plasmocine consists two types of antibiotics. Okay, so the first one is to block the protein synthesis, and the second one is to stop the DNA replication. Okay, so now this particular plasmolysin, all right, again, if you see, it is very much faster, all right, it can rescue your cells basically within 2 weeks, and there is no toxicity or no to little toxicity on your cell cultures. Okay, and this plasmolysin comes in two types of packaging. So we have the we have the prophylactic, which is used to prevent the contamination, while the other is used as the treatment. So basically you have two types of packaging. If you can see here on the picture, you have the treatment and the prophylactic. So this particular packaging with the red tube here, so it is used to prevent, okay, it is used to prevent the contamination, and the treatment is used to eliminate. All right, so um if you see why you should get plasmolysin, because one of it is responsible, it can actually eliminate 95% of your cell culture contaminants. All right, it is effective both for extra and intracellular. All right, it does not interfere with other antibiotics, so you can use it together. All right, and like what I said, you can use routinely, and again, it is actually very famously, it's been a very famous product in a lot of research and publication. All right, so Plasmocure is basically the second line anti-mycoplasma reagent. So if the first one we saw plasmocine, so plasmocine is the first line defense. So if there is any mycoplasma that survives, so you can also look into Plasmocure. So Plasmocure is more like the second line anti-mycoplasma reagent. So anything that is resistant to plasmocine, so this Plasmocure will be able to remove it. Okay, so now if you see here, you can see the chart. So we have the Plasmocure plasmocine. So whatever plasmocine cannot be doing it, we have the Plasmocure to help it. All right, so this is on the cultured cell, culture death, mycoplasma regrowth. So you can see all these, how they are inhibiting the mycoplasma regrowth of it. Okay, so now this particular product, okay, this particular product, if you can see here, these are the treatments. So basically it rescues within 2 weeks. All right, little toxicity to your mammalian cells, and for the regrowth of the mycoplasma. So it has the extremely low regrowth rate. Okay, so you can see how's the efficacy and the resistance. So basically it is very much okay, so efficacy is much more higher and resistance is very much lower. All right, so this is actually the new kit that has just come into the market. All right, which is the Strip. Okay, So this particular microscope is actually very simple and very rapid detection kit. It is something like the pregnancy test, right? If you see the kit is something like that. But now you can use such kit to just detect the mycoplasma in your cell. So the technology that is using is the detection method. It uses the isothermal PCR, okay, of the 16S rRNA gene. So basically they all you have to do is you just have to mix. All right, this is the sample preparation. So you just um, you have your prepared sample, which means your cell culture supernatant, so you can have that. And then you have your reaction buffer you insert in with your reaction mix. All right, you incubate it at 65 degrees Celsius for 40 minutes, and then you add the migration buffer, you mix it, and then you directly add into the cassette. Okay, so it is very direct, because if you have two bands, so even if you have a faint band, it is still showing positive for your for your mycoplasma. And if you just have one band present, all right, means it is negative. So it's pretty much direct. Okay, so this particular test, it takes complete lab in like around 1 hour, and you just need about 15 minutes of hands-on time. So it's very direct. You can literally interpret the results within 2 to 5 minutes. Okay, and this particular kit, it is also can detect Mycoplasma species, as well as the Aculoplasma species as well. So we have the two species that actually it can detect, right? So now let's move on to bacteria. Now, bacteria is basically large ubiquitous group of unicellular microorganism. So it has few micrometers in diameters, and they tend to have they have variety of shapes, basically circular, rod, right? So they are also known to be the common encounter of contaminant in cell culture. Now, though you can detect them using a light microscope, but usually bacteria can be easily mistaken for cellular debris. So when it is in the initial stage, this is what a confusion can take place. Okay. So now we know when it comes to bacteria, there are two types. We have the gram positive as well as the gram negative. So here are some of the examples. So how can we detect to make sure it is bacteria and not a cell debris? All right. So now we know that cellular debris is much more smaller. Because cellular debris is much more smaller than the eukaryotic, so we tend to view them under the microscope, trying to see if it's possibility of bacteria contamination. So if you are using a phenol base cell culture media, then you could directly see there will be color change. Okay, it changes the color to be cloudy. So the moment there's a cloudiness in your in the media, yeah, you have to discard, right? So let's look into the source of contamination before we move on how to detect them. So the source of contamination, again, your lab operators, okay, basically the researchers who are operating that particular cell culture work. All right. Dry water bath, incubators, glasswares, they can also come from environment and also subclinical infection, depending on what kind of cell or cell lines that you're using. Now, we know for bacteria, it is always the way to eliminate is using antibiotics. So what we have here is, it's not one type of antibiotics, but it comes with a formulation of 3 antibiotics. So these 3 antibiotics, it's not only effective for bacteria, but it's also effective for mycoplasma and 2 classes of bacteria. And you can actually use this as a routine addition to prevent contamination. And the good news is you can always combine the normicin with your pen strap. Okay, and it has very minimum cytotoxicity against the cell. So this is the product information. So we have in two packaging size. One is 500 mg, another one more is 1 gram. All right. So this is Normocine. And now Normocure, for example, like this is to encourage. All right. So like something like a second line defense, I would say. So again, if your pen strep and Normocine, so if you have any bacteria that still strive the growth, despite of using pen strep and Normocine, now you can use Normocure as well. So they are actually very much effective against gram positive and gram negative bacteria. So this is the gram positive and negative, which I've earlier I've just shown the particular chart. Okay, so usually what they can do is they can eliminate at least 99% of bacterial contamination with minimum cytotoxicity against cell. So Normocure becomes the second line defense. Then we have Primozine. Now, Primozine, it's also antimicrobial agent. Okay, it still kills, but now it gives a broad spectrum because it's able to do killing with for Mycoplasma bacteria and fungus as well. Okay, and it is non-toxic to primary cells. So if you see Primozine, it consists of 4 compounds. Okay, now 3 compounds will work in blocking the DNA, protein synthesis, and also mycoplasma. And the 4th compound, it tend to eradicate the fungi. So these are the 4 compounds that can be used. So you can even use the Primozine as one, all right, to prevent your cell culture from getting infected by mycoplasma bacteria or fungus. Now, Primozine, you can actually use them with your primary cell or any human tumor-derived cell lines, stem cells, or also embryonic cells. So we have publication always. You can go into this particular web page. You can see publications on Primozine as well. All right. We have also 2 kind of packaging, 500 mg as well as 1 gram. Now, so much so for we have seen for Mycoplasma and bacteria. Now, endotoxin is also part of the contamination, so we can't leave endotoxin out. So, endotoxin, known as the polysaccharide or lipoglands, basically cell wall component of the gram-negative bacteria. Okay, so they are the potent inducers for inflammatory response in both in vivo and also in vitro. Okay, so now if you see lipopolysaccharide LPS, so this is the structure on my right. If you can see the LPS basic structure. Now this endotoxin, they are approximately 10 kilodalton in size. So but they are readily form large aggregates, and they can go up to 1000 kilodalton. Okay, so they are amphipathic molecules, and what they do is they carry a negative charge in solution. So because of the hydrophobicity, they are likely to have strong affinities for other hydrophobic materials like plastic product in your that you use in your lab. All right, so for endotoxin, the main reason the main source where you find them is at the carryover contamination from your plastic wares. So for example, your beakers, stir bars, or any other labware, okay, that is used. So that is where you will find the source of endotoxin. Okay, so solution and reagents may be sterile. You think they are sterile, but sometimes they will be still having components, okay, components of bacterial, that is the LPS. Okay, so monitoring for the presence of endotoxin in cell culture reagents is extremely crucial. All right, so now, so because of that, we are going to see on the impact and source. Okay, now let me bring you through the impact. So endotoxin presence can actually actually can significantly impact both in vitro and in vivo. This is like how I've explained earlier. All right. Although the effect of endotoxin vary according to the concentration and cell type, so it is shown that this particular molecule can alter the cell morphology. Okay, not only the cell morphology, but also the proliferation and transfection efficiency. Okay, now when there is the presence of LPS, it will automatically enhance the production of inflammatory cytokines. So some of the cytokines are, for example, the TNF alpha, interleukin 6, interleukin 8. So these are some of the inflammatory cytokines. Okay, and what can they do? So once this has been activated, sometimes it is able to cause pyroprotic cell death. Basically, it causes cell death. Okay, all right. So now, what are the source of contamination? Like what I said, usually comes from your lab waste. All right, so make sure your water, when you use water, it is highly purified water that is used to prepare media and solution. Use a clean glassware. All right, to destroy endotoxin, you can actually heat the glassware at 250 Celsius for more than 30 minutes or at 180 for 3 hours. Right, so this is one way to actually destroy your endotoxin, and you can always ask your manufacturer to make sure that whatever media you buy, whatever cell lines that you buy, they are actually free from endotoxin. All right, and example, your your culture dish. All right, so your T25, so all these particular culture dish, you can always ask if there is certification for endotoxin free. Okay, so these things, they must have the certification for the endotoxin free and also absence of pyrogenicity. So you can always check these from the company that you buy, from the vendor you buy. So you can always ask if this particular product is free from all these. You can always ask for certificate for them. All right, so how are we going to eliminate them? Okay, when it comes to endotoxin, now this is actually a colorimetric cell based assay, right? So it is a HEC Blue LPS detection kit too. All right, so now this kit basically relies on the production of SEEP, which is actually the secreted embryonic alkaline phosphatase reporter gene. So this is the SEEP here. All right, so this is the kit. So basically it can detect as low as 0.01 EU per mil. Okay, and one kit, when you purchase one kit, it can go up to 500 tests. So you can perform 500 tests based on this detection kit. Okay, so what is the principle, or what is the protocol? So basically all you have to do is to add the endotoxin containing samples, okay, to the HEC Blue 4 cells cultured in medium. So you just have to add them. You incubate overnight, okay, 37 degrees Celsius, 5% of CO2. Now, if there is present, so you'll have the purple blue. All right, the color will tend to change. Okay, so when do they change? When you add the Quanti Blue solution. All right, so once you have these, you add in the Quanti Blue solution, you incubate them to about 1 to 3 hours, then you will start measuring. You can actually measure, or you can also see them in naked eye. You can see them changing colors. So if it's a pink, then it is this particular cell line, or the cells that have been cultured in that particular flask. All right, it is free from endotoxin. So if there is a positive, then you will get to see the color changing from purple blue. All right, so what are the advantages of using okay, this particular kit. So now it is able to this particular kit actually, it is alternative to the Limulus amoebocyte lysates, which is the LALSA. All right. Now you can purchase this particular kit and you can always freeze them. All right, and then you can thaw them and you can reuse back. So it is not that you once you have opened up you have to use it all. It's no it's not like that. All right. And unlike the LALSA, now this particular hack blue, okay, it is based on the LPS detection and it does not give you false positive, okay, result if the sample contains 1,3 beta D-glucan. So this is also makes it more in an advantage prospect. Okay, now you can use all type of biological samples. All right, so these are the biological samples you can use to detect LPS. You can use medium, serum, chemical preparation, or even vaccine adjuvants. Okay, and it's very much easy to visual because you can even visual to your naked eyes, or you if you have a plate reader, so you can also visualize from there. So it is actually changing from medium color of pink to purple. So this is the product information. If you see, it comes with the detection kit. There is a selection. You also have a standard with it. Okay, and two other antibiotics, which is the Nomocine as well. So this is the kit, the description and the quantity of it. Now, lastly, let's look into fungal. Now, when it comes to fungal, the most common one, okay, would be yeast. All right. So fungal can actually exist as round or oval bodies. All right. Or they can also form a hyphae. Now, not only fungal, but molds can also can cause contamination. Okay. Now they become a bit hard to remove it because they spread via spore mobility in the air. Means they can always spread through air once you open up the culture. So some of these spores, they are dormant until a suitable condition is available, and then they actually grow back. So how are we going to detect fungal contamination in a cell culture? All right. So now when it comes to yeast, all right, so yeast is the smallest form of fungi. Approximately they range between 3 to 10 micron, okay, the size of it. So usually you usually can be observed under a light microscope. All right, for yeast you can actually observe, but when it comes to hyphae, it can be it can be detected by the naked eye or light microscope, depending on the size and the growth stage. All right, so if it's very highly contaminated with fungus, so basically you'll start seeing hyphae. So literally you can see them with your naked eye. Now when it comes to mole will have a floating colonies form in the surface. So when you have moles or you have hyphae or yeast, it can be because we wouldn't know who is producing a dormant spore. So you wouldn't want to open such culture disc, and you want to avoid spreading the spore. So this is the most important. So when you see such, you need to actually discard that particular sample. Now, how are we going to eliminate is by using fungin. So fungin is actually used, you can use it for yeast, hyphae and molds, and it can rescue the particular cell culture within 5 to 10 days. Okay, it is also considered quite safe because it has a very minimum toxicity towards the mammalian cells. All right, and you can also use this as a prophylactic treatment. Okay, now this particular fungin, it is an excellent alternative to instead of using the amphoterine B antimycotic, all right, you can use a fungin. Okay, now you can use either low or high concentration for routine preventive, especially when if you think that your culture is contaminated with candida or aspergillus, so you can actually use them in a relatively suitable concentration, which you can actually optimize as well. Okay, now a good news is you can always add them into media, and you can use as an alternative to penicillin and streptomycin. So this is like an alternative for your pen strep. So when we come to fungin, we have two kind of packaging. So we have the 75 milligram and 200 milligram. All right. So you can either choose 5 tubes, 1.5 mil, or 1 tube in 20 mil. Now, so this is the conclusion. So basically the summary of all antimicrobial agents that I have discussed earlier. All right. So we have for mycoplasma, bacteria, endotoxin, and fungi. All right. So these are some of the products and their description. So microbial contamination is actually a major issue in cell culture, but there are actually a range of a range of procedures which can be adopted to basically you want to prevent and eliminate contamination. Now, Invivogen has done an excellent job, or excellent, they provide an excellent solution to overcome this contamination. All right. So, Invivogen cell culture contaminants, okay, detection. They have prevention and elimination products. They are basically able to help you solve at every stage of microbial infection. So this is good news from Invivogen. So if you have mycoplasma, we have arranged to detect, we have arranged to prevent and also eliminate. So is for the rest of the microorganism contaminants. So with that, I end my presentation. So if you all have any questions, please type in the questions in the question box, and we will get back to you through email.